So here we are to celebrate. We're here to celebrate science and science excellence with a wonderful symposium title, From Tree Genomes to Future Forests. And I know, having heard the presentations already, that that story will be very clear as we move through the program. Jerry, specifically to you, we hope that you're going to be able to listen to the presentations, other than your own, of course, and see the incredible impact that that first pivotal study has made on the field of forest and forestry. So please also enjoy. To get us started, I have the privilege of welcoming the chairman of the Marcus Wallenberg Foundation, Mr. Marcus Wallenberg, to say a few words. Marcus, the floor.
Thank you. Good morning. Thank you, Mike, and congratulations again, Jerry. Great to have you here. Great to have everybody here today. And as Mike said, a lot of people online, which is great. We also have, I think, 26 young researchers, which we are very, very happy for, because this has been the aim of the foundation for a long, long time, to try to get even more young researchers engaged in this industry in general.
I think there's a nice overview that'll help you, I think, put everything to come in context. So when we do tree biotech or genetics, generally, we're using natural genetic diversity. We may amplify it by making hybrids and putting them in unique environments. And then we're putting them in the field in some structured way so we can see their genetic value versus their phenotypic value, their environmental influences. And that's mostly how we do breeding, right? We see, are the trees growing fast? Are they disease resistant? Do they have good wood properties, and so on and so forth? And then increasingly, today, and you'll hear some great talks about this coming up, we can use the genome. We can put tens of thousands of markers throughout the genome and use that as an index. We train that empirically, meaning we go to those same field trials and correlate the genomic information with the phenotypes. So it's not based on knowledge of the genes. It's based on just having so many markers that we can characterize the genome in general. So, and this technology, because of the sequencing revolutions that have happened, computation, that is pretty mature technology. Anybody can do it today. It's really a matter of economics. So that's very exciting. That's the left side of the panel in front of you. On the right side of the panel is what I'm calling functional biotechnology. It starts with knowledge of the genes and the pathways and what they do, so we can tweak them in an informed manner. We can make a new kind of gene that's expressed a little differently, put it in. It can come from the same species or a different species, or we can edit the gene, change some bases to make it do something we want it to do. And that, and you see this ideal of these things coming together. This is like what biotech companies do. They're doing conventional breeding with genomics, but they're also adding in these particular genes for particular traits. And in forestry, really can't do that yet. It's very, very rare worldwide to be able to have the functional side come in. So we'll talk about this cleavage and what we might do about it as we go.
I wish I would be around for the next 50 years. Biology is going to change the way we live, the way we manufacture, the way we heal ourselves, where we now know that certain traits, certain behaviors occur at the tissue level, not on the whole plant level. And with that, we can identify not only networks of genes that are expressed at the tissue level or the cell level, but we can find promoters that are associated with that network of expression. And then we can take those promoters, and instead of doing constitutive overexpression of a gene on the whole plant, we can use that tissue level or cell level promoter to only turn the gene on in the tissue at the time we want it turned on. And this will revolutionize how we approach transgenics in the future and CRISPR-Cas9 modifications. A single change in a nucleotide, ATGC, can lead to an amino acid substitution that then can lead to a phenotype that's valuable to foresters deploying this ultimately in the field. From this, we associated every phenotype we could measure and found 35 genes that went into invention disclosures. It turns out that there's a single gene, it's a protein, that causes a hairpin or like Velcro structure. So in rough bark trees, the Velcro held the dead cells next to the dead cells, and that leads to rough bark. But the smooth bark, the protein has a single amino acid substitution that causes the protein to flatten, and therefore neighboring cells slough off. That is wild type in humans. The reason we have smooth skin is that that exact same protein, the exact same protein in poplar causes a disease in humans called keratoderma, where the protein has mutated and forms hairpin. So I think we're going to see a lot more of this high throughput phenotyping, not only in the greenhouse, but in unmanned aerial vehicles, flying over our forests and characterizing behaviors on broad swaths of land in nature. I've had the benefit and privilege of working with dozens and dozens of hardworking scientists who elevated my science greatly, and I wouldn't be here today without them. As early career scientists do the same, find those bright, hardworking people, collaborate with them, and you too one day might be standing here on this stage. So thank you very much.
Researchers can now create new, resilient varieties much more efficiently and much faster. We could make plants more resistant to disease and drought. We could allow farmers to use fewer pesticides and protect harvests and yield. These are award-winning and climate-friendly innovations. If we would use them, it can strengthen our competitiveness, it can make us more resilient to climate change, and we can also help to protect the environment. Because Europe is at a turning point. Modern science was born here. We have tremendous knowledge, experience, and talent. We have all the conditions that would enable us to continue being a scientific and economic powerhouse.
Tree breeding has evolved quite a bit in the last 30 years. We have well-developed methods of flower induction, for example, to allow going from seed to seed in 1 year in the case of eucalypts. We can generate large full-seed families, clonally propagate almost all genotypes, and use advanced field trial designs for measuring traits of interest with very good accuracy. For applied breeding, we have moved from trying to discover individual gene to capturing the full aggregate of the genome effect like quantitative genetics has historically done, but now using genomic data. To carry out genomic selection, we use SNP markers. Single nucleotide polymorphism is the most common variation in genomes. For eucalypts, we have developed operational SNP platforms used worldwide for the 20 or so eucalyptus species by more than 50 companies or research institutions. Genomic selection is like investing in index funds. It's cheaper, simpler, humbler, and generally more efficient. Instead of trying to pick single winning stocks or genes, we breeders simply buy the whole market, or better said, the whole genome effect. Now, many financial studies show that this tends to be more efficient on the medium to long term, and we believe this is the same case in tree breeding.
And as Dario pointed out as well, genomic selection models will help us to do predictions for disease resistance. And we hope to achieve a tool for yield protection so that we have sustainable forests.
Then, and the last, for me it's important that we continue to train, and that to train differently tree breeding and genomics, and in order to update skills in genomics and breedings, and also to be able to integrate new jobs in our breeding sector to integrate this genomic technology.